All right, good afternoon, everyone, and thank you so much for joining today. Welcome to the Space at Hopkins Virtual Symposium. My name is Carolina Nunez, and I am the Space Fellow for Space at Hopkins and a PhD candidate in the Krieger School of Arts and Sciences, Department of Physics and Astronomy. It's my great pleasure to be able to moderate this event today. On behalf of the Space at Hopkins team and Professor Chuck Bennett, I would like to express just how thrilled we are to be able to host such an exciting program this afternoon. Today's symposium aims to showcase the diversity of space-related research involving Johns Hopkins researchers across the university. Space at Hopkins is an umbrella organization connecting the array of Johns Hopkins University divisions, departments, and collaborative institutions in their common pursuit of civilian space research. We aim to highlight and encourage the breadth and depth of Johns Hopkins University's efforts in space-related activities and to foster collaborations. For you students calling in today, we hope to provide a glimpse of the space-related research opportunities that are available across campus. Since its initiation in 2016, Space at Hopkins has hosted a variety of events, including symposia, special workshops, informal social hours on several Johns Hopkins campuses. We've had the pleasure of hosting astronaut Kate Rubins, who collaborates with Johns Hopkins biomedical researchers, and sponsored 2 conversations in medicine events with flight surgeon Dr. Jonathan Clark and Dr. Andrew Feinberg. We also held a session where Johns Hopkins students met with Northrop Grumman leaders about industry careers. These events are meant to foster informal discussion across all university divisions and to connect the university with the external space community. Normally, we would host a reception following today's symposium, and we regret that this is not possible given the current circumstances. However, we strongly encourage you to get in touch with one another. Introduce yourselves, ask follow-up questions. Please continue our tradition of discussion and collaboration. So Space at Hopkins oversees the Maryland Space Grant Consortium, connects with the cooperative agreement between Johns Hopkins University and the NASA Goddard Spaceflight Center. We also host the webpage for the undergraduate minor in space science and engineering. A key component at Space at Hopkins is the annual seed grant program. So each year, Space at Hopkins awards seed grants of up to $25,000 that enable Hopkins researchers to test new ideas and provide an initial boost for them to obtain external funding. We especially encourage interdisciplinary projects and collaboration across Johns Hopkins divisions. In addition, all Space at Hopkins seed grants involve undergraduate students in research opportunities. All Johns Hopkins University professionals who are eligible PIs on external grants are eligible to apply for the seed grant funding, and proposals for the seed grants are due in the spring of each year. We'd like to take a moment here to congratulate the 5 new 2020 seed grant recipients representing the Applied Physics Laboratory, the Krieger School of Arts and Sciences, the School of Medicine, and the Whiting School of Engineering. Further information about these past grants can be found on our website. spacestudies.jhu.edu. Today, we are thrilled to showcase the 2019 seed grant recipients and their research at this symposium, in addition to featured talks by Dr. Jason Callery and astronaut Reid Wiseman. We will be taking questions throughout the event via the Zoom Q&A feature. We encourage you to submit any questions you have during the presentations. So Dr. Matt Collins, Deputy Director of the Maryland Space Grant Consortium, will be monitoring the Q&A and pose your questions to the speakers as time permits. All right, so I'd like to turn the stage now to the Seed Grant Showcase. We will have 5 speakers today, all recipients of the Space at Hopkins Seed Grant Award in 2019. Once again, for those of you just calling in, we encourage you to ask questions via the Zoom Q&A feature during the talk, so they're ready for Matt to moderate following each presentation. So our first Seed Grant speaker today will be Dr. John Appel. Dr. Appel is an associate research scientist in the Department of Physics and Astronomy in the Krieger School of Arts and Sciences. Dr. Appel is a detector expert in the field of cosmology and a leader within the collaboration of the Cosmology Large Angular Scale Surveyor, which is a polarization-sensitive cosmology experiment.
Hi, my name is John Appel, and I am a research scientist in the Johns Hopkins Physics and Astronomy Department. Today, I will be talking about the V-CubeSat project funded by Space at Hopkins grant. I will begin by introducing the project's science goals, followed by a brief overview of the undergraduate students involved and the mission concepts they developed. And I'll end with a brief description of the proposals catalyzed by this Space at Hopkins project. This figure illustrates the history of our universe. On the right is the present day, and if we look back to the left, 13.8 billion years, we reach the moment of the Big Bang and the beginning of our universe. With the V-CubeSat project, we are interested in studying the early universe, in particular, the periods of the ionization and inflation. The ionization starts when the first stars ignite and the light reionizes the neutral hydrogen that fills the universe. Inflation is a period of exponential expansion moments after the Big Bang that inflated quantum scales to macroscopic distances. We can study both these two periods by measuring the cosmic microwave background, or CMB. The CMB photons we see today were part of the primordial plasma that filled all of space. The expansion of the universe cooled the plasma to a temperature where protons and electrons combined into neutral hydrogen. At this point in time, around 400,000 years after the Big Bang, the CMB photons decoupled from matter at a temperature of roughly 3000 Kelvin. The continued expansion of the universe cooled the photons to the 2.7 Kelvin blackbody temperature we see today, with their intensity peaking in the microwave frequency range. We study the periods of inflation and the ionization by measuring the characteristic polarization patterns they imprint on the CMB. Plotted on this slide is the state-of-the-art full-sky CMB polarization anisotropy measurement by the Planck satellite, released in 2018. Note how the amplitudes of the anisotropies are small on the level of microkelvin. The goal of the V-CubeSat is to improve on this measurement by increasing map sensitivity and reducing systematic errors. The project targets V-band, which is a microwave band near the polarization foreground minimum as shown in this plot. Galactic synchrotron dominates at lower frequencies and thermal dust at higher ones. Also note that V-band is not accessible from the ground because of an atmospheric oxygen emission line. So this type of measurement has to be done from space. A major component of the project was to involve undergraduate students across JHU to develop the mission concept. 8 undergraduates were hired across multiple departments, including physics, computer science, and engineering. We had a diverse group of students, including women and underrepresented minorities. The students developed a thermal simulation of the CubeSat. The thermal model shown here indicates that with 4 radiation shields, we can passively cool our detectors to 63 Kelvin. Cooling the detectors is important to improve sensitivity of the instrument. They also characterized the orbit and scanning solution that would allow us to map the entire sky every 6 months, while keeping the detectors shielded from solar and Earth thermal radiation. As shown in the diagram, the CubeSat axis points away from the Earth and Sun, and the instrument rotates about its axis roughly once a minute. In this way, each detector scans large circles on the sky as the satellite follows a sun-synchronous orbit with about a 2-hour period. This figure shows the parameters that define our low Earth sun-synchronous orbit, which is also commonly used by commercial satellites. These and other concepts developed by the students were the foundations for a proposal to NASA's Astrophysics Science Small Sat Study Program. Additionally, the project was modified into an ESPA small-sat mission concept called CRIS, presented to the Goddard Spaceflight Center Pioneer Panel in July. We also plan to propose to develop detectors for a Ku-band small-sat that will characterize the synchrotron galactic foreground at frequencies near 15 gigahertz. Thank you for your time. I will end here with a video animation made by one of the students depicting the V-CubeSat's orbit across the solar system.
Thank you, John. I'll pass the floor over to Matt for the Q&A.
Okay, thank you, Carolina. John, so the first question, we know you can't predict the future, but one might think of a CubeSat as being like the entry level of designing a satellite. I don't know if that's a fair attitude, but can you give a feeling for how many stages you might expect to have to go through between the initial concept that you first came up with and the work that you've done now and when a satellite might actually launch?
Yeah, can you guys hear me? Just to check.
Yep, we hear you just fine.
Yeah, I mean, so this is part of the learning process to see what steps are required, but definitely the small sat provides the opportunity to get your feet wet and do a space mission that maybe is not the size of a mid-range, and so it provides a lot of valuable experience. One challenge is that to really do cutting edge science, you really need I mean, a lot of these measurements have been made by billion-dollar satellites. So to really get to the frontier of science, it's tough to compete with a small sat. But I think it is important to develop a plan in which maybe you can use some of these small sats to kind of build your program and work around the edges where you can make progress on the science, and then you build the experience, and then in the longer term you'll be ready to have a successful large mission.
Okay, thank you. Another question. Could you tell us a little bit more about the type of detectors that are used for the type of measurement the satellite will make?
Yes, so these are radiometers based on Hempt amplifiers. So these detectors are very similar to what was used in previous satellites for measuring the cosmic microwave background, like WMAP. One advantage of these type of radiometers that measure the absolute power in these microwave frequency bands, but are also sensitive to polarization, is that they operate down like optimally at 20 Kelvin, but you can also operate them at 60 or 100 Kelvin. And so it simplifies the engineering problem. We would love to put in space TS bolometers, for example, but those operate cryogenically below 500 millikelvin, and doing cryogenics in space is expensive, and so you can't do it at the moment. You can't do it with a small sat. So that's one advantage of targeting this type of mission that uses Hempt-based radiometers, that the cryogenic challenges are something we can tackle right away.
Okay, thank you. That is all the questions we have for now. So I will pass the virtual floor back to Carolina.
Thank you. Thank you very much. All right. So our next Steve Grant speaker today is Dr. Marco Chiaberge. Dr. Chiaberge is an associate research scientist in the Department of Physics and Astronomy in the Krieger School of Arts and Sciences. He also serves as a senior European Space Agency Aura astronomer at the Space Telescope Science Institute. And Dr. Chiaberge is an expert, is an expert on active galactic nuclei and supermassive black holes. He's a member of the Advanced Camera for Surveys team for the Hubble Space Telescope.
Hi, everybody. I'm Marco Chiaberge, and I'm very happy to talk to you today about this project on behalf of my team. My team is made of people from different schools and different institutes, different departments at Hopkins. We have biologists, physicists, astronomers, engineers, and a couple of external collaborators from the medical field too. Today we're going to talk about the effects of microgravity on articular cartilage and how we can counter those for long-duration human spaceflight. So spaceflight exposes the human body to microgravity and radiation. The human body is not prepared to experience such an environment, especially for a long period of time. If you just go to Hubble for a couple of days to fix it, as you can see in the right-hand side of this slide, then you're probably going to be fine. But if you stay for longer periods of time, for example, on the International Space Station, or in the future for longer spaceflight to Mars, for example, then the effects of microgravity become substantial. Effects on muscles and bones have been studied for many years, and countermeasures were developed, and they are continuously updated. Here you see an astronaut riding a stationary bike on the ISS, for example. One aspect that has been poorly studied is the effect of microgravity on articular cartilage. Cartilage is a very important component of our body because it covers the bones at the joints, and it allows the joints to move smoothly, and also bears the whole human weight. So it's very important. If cartilage is not working as it's supposed to be, and you have osteoarthritis, then you cannot walk, you cannot move around. So there is evidence that immobilization and reduced loading lead to changes in the structure of cartilage, and experiments in suspended mice to simulate microgravity environments show that degradation happens after only a couple of weeks. So if astronauts are subject to the same cartilage degradation as seen in some lab experiments, then even traveling to Mars may be impossible, because the astronauts get there, and when they get to the surface, they cannot walk around because their joints are not working anymore. So we must develop countermeasures. Our hypothesis is that plyometric exercise, so jumps, may be helpful to reduce damaging effect of microgravity on articular cartilage more than other type of exercises, for example, riding a stationary bike or running on a treadmill. This is for mainly two reasons. One is because the type of mechanical loading during plyometric exercise is very intense, but very short in time. And this is shown to stimulate cartilage. This has been shown to stimulate cartilage. The other reason why we think this is going to be effective is that this type of exercise stimulates secretion of hormones that are important in cartilage health. So how do we test this? Well, we need to do mice model experiments. And so we need a mouse to jump, as you can see in this little animation. The problem is that currently there is no apparatus that is available to force mice to jump in a controlled manner. And so that was the goal of the Space at Hopkins seed grant, to support engineering and building a prototype to show that we can do that. We can make mice jump, and we can do it in a controlled manner. And that will enable experiments. So the system we came up with works in this way. We have a mouse in this lower platform. The mouse gets shocked. Mouse jumps. Then a gate closes. We have a sensor that tells us that the mouse is not there anymore. And then the platform is elevated, and the platform where the mice is lowered, and the gate is opened again, shock comes in, and the cycle goes on for as many jumps we need. There's also a light that is turned on right before the shock comes in, so the mouse can learn that when the light is turned on, they need to jump. This is the design of the prototype, the MJ2020 prototype 1. I can't get into the details, but you can see in the center the platforms and the hatch, and on the sides you have the motors that move the platforms up and down. This is a picture of the real thing in the lab. When we did this test, the apparatus was commanded by Anderson, who was at home. So it can be also controlled remotely. We also had a mouse inside, which is not visible in this picture, but it will be visible in this video, in which we show the test that we did in September to show that the system works. And the mouse is at the bottom, and there it goes. So in this case, the mouse learned immediately that when the light is turned on, they needed to jump. I'm going to show it one more time. There you go. So the system works. This was the goal of the project. We found a way, and it works. So my conclusions are that cartilage degradation in long-duration human spaceflight is a concern. The hypothesis is that plyometric exercise is useful to reduce damaging effect of microgravity on cartilage. And we needed a prototype. We needed to engineer a prototype to make mice jump. And in the future, using what we learned from building this prototype, we will be able to build a bigger apparatus to train a number of mice simultaneously, and start doing experiments to implement this for, in the long term, use it for astronauts. Note that there may be relevance for rehabilitation and treatment and prevention of patients with osteoarthritis. So this is also a project that may have implications for the medical field. Thank you very much for your attention, and I'm happy to take questions.
All right, go ahead, Matt.
Okay, thank you. So we do have a couple of questions coming in. So first question is the benefit of this plyometric exercise mainly preventative, or in your opinion, do you think that plyometrics could actually help regenerate cartilage?
I cannot turn on my video for some reason, but I can answer that. Maybe both. There is actually there is evidence from one paper, from one study that was published by a Finnish group. They did an experiment on a group of women with mild osteoarthritis, and they trained them for a number of weeks doing jumps. And they saw that the cartilage was getting thicker. So that it's possible that there's a benefit also for people with mild osteoarthritis. Probably not the worst cases, but the original idea we had was basically to train, to find the best way to train astronauts to prevent any damage during long duration spaceflight.
Thank you. And a related question, what is the concept that you have for getting appropriate joint loading while jumping in microgravity? Basically, how do you envision translating plyometrics into space?
Yeah, that's a great question. So let's see if I can show you, if I can share my screen for a sec. I can show you a picture of, I hope you can see it. So this is a machine that is routinely used in rehabilitation center. It's a plyopress. And the way it works is basically using weights on Earth. You can imagine doing something very similar using, for example, resistance bands in space, because you can't really take advantage of anything that weighs something in microgravity environments. But there are ways, so it's not something that it's impossible to do in space. This is what we have in mind for the future.
And a related question, how does this compare to current exercise regimes that are used on ISS?
Yeah, that is one of the biggest questions. So when we actually will be able to use a bigger machine that we hope we're going to build soon, because now we know how to do it, we will be able to do experiments with samples of mice that are trained in different ways. and that are also suspended to simulate the effects of joint unloading. And so we will be able to compare the effect of this different trainings on the health of their joints. And that's going to be the answer. One of the biggest questions is the type of training that the astronauts are doing right now on the space station enough to prevent cartilage damage, or do we need something else? We don't really know, and it's a very important question to answer.
Thank you. Taking a step back, could you say something about why mice are a good proxy for human physiology?
This would be a great question for my collaborator, the biologist fan. I think that mice are widely used to test all possible things. Like even the coronavirus vaccine has been tested on mice. So you can imagine using other animals. But I think that biology has established that mice, and especially specific type of mice, there are very different ways. Now mice can be engineered to be more closely related to the way the human body is made. So starting with mice is usually what is routinely done. We can't really imagine doing anything different from what the standard protocol is right now. I hope that answers the question.
Thank you. So you mentioned coronavirus in your answer to that question, and the next question concerns that. The question is, what impact did the COVID pandemic shutdown have on the timeline for your project and on how much you were able to accomplish?
Yeah, it was tough because also most of the work, the design and really building the prototype was done by our great student Anderson for Lanetta, who did really great job. And not having him on site was a big problem. So for some time he had to work at home with the parts and bits and pieces. The design was done remotely, so that was easy. And also one good thing is that the machine, the apparatus can be controlled remotely. So as you saw in the experiment, Anderson was at home, and we had technicians in the lab. And so we were able to handle the changes, but it slowed down the process a little significantly. We had one engineer, Steve Hope, that had to do a lot more work than we anticipated. And so that increased the cost significantly. But we managed to finish it, which is great, and it works, which is even better.
Indeed. So we're getting up close to our time limit here, but I think we have one more question. It refers back to one of the earlier ones. The question is, would plyometric exercises be effective if cartilage degradation has already occurred to a severe degree in astronauts? Or would something else, like stationary bikes that have possibly less stress-inducing nature, be more appropriate at that point?
In just short answer is we don't know. What is likely is that just riding a bike does not help cartilage to get better. It's actually very hard to heal cartilage when it's already damaged. Cartilage is a very special component of our body, so it doesn't really heal. As you all know, if you get a meniscus tear, surgery is a very likely outcome because it doesn't heal by itself. So it's our work is aimed at preventing bad things to happen before fixing it. But there are a couple of experiments that we have in mind that we can do with athletes, healthy athletes, that will help us to see if this can stimulate cartilage at all levels. But I don't think that can be used if the situation is already above a certain level of degradation.
Thank you. I'm going to push the time to make room for this last question because it's very Space at Hopkins specific. Where can those interested in space medicine or space physiology go to learn more about this domain? Are there any literature, professional societies, or other resources that you would recommend within the Hopkins sphere or otherwise?
Okay, well, that is a perfect assist for me to advertise our new seminar series that we are about to organize. And this is, it's going to be called Seminar for Space, Biology, and Exercise. We're planning on having a couple of events per semester. You'll hear more about this on the Physics and Astronomy website, and we will advertise this. I think that Space at Hopkins is a great source. I don't have any specific recommendation. I just really, if I may, I just really want to thank Space at Hopkins for giving me the opportunity to start this project and allowed me to build this incredible team of collaborators. And we have our Mark Schellheimer, our human spaceflight expert, Cheng-Ming Fan, our biologist, Steve Me and his team at the IDG. We wouldn't have been able to do this without them. And this is just the beginning. So yeah, I would keep an eye on the things that we are about to organize. Mark Schellheimer at the School of Medicine also has very interesting things on his website about human spaceflight. So those are the things that I would check out.
Okay, thank you very much. And with that, I will pass the virtual stage back to Carolina.
Thank you all.
Thank you, Marco. I think I can speak for the Hopkins team and say it's our pleasure. All right, so our third Seed Grant speaker today will be Professor Jamie Guest. Professor Jamie Guest is an Associate Professor of Civil and Systems Engineering in the Whiting School of Engineering. He also has a secondary appointment in the Department of Material Science and Engineering. Professor Guest is the leader of the Topology Optimization Group at Johns Hopkins. He is also Associate Director of the Johns Hopkins Center for Additive Manufacturing and Architected Materials, and Associate Director of the Center for Integrated Structure Materials Modeling and Simulation.
Hi everyone, my name is Jamie Guest and I'm faculty in the Civil and Systems Engineering Department in the Whiting School of Engineering. Before I tell you a little bit about our 3D woven carbon nanotube composites, let me just acknowledge my collaborators here, Professor Tim Weiss, Christina Ramani, who is a senior, and Hak Young Lee, who is a PhD student. The students, of course, did all the work and did great work and deserve all the credit for this project. So the motivation for this project was quite straightforward. Spacecraft, and particularly launch of spacecraft, is very expensive, and mass is one of the primary drivers for those launch costs. And just to give you an idea of how large that is, it costs around $10,000 to place a pound of payload into Earth orbit. It costs about 10 times that or even more to send that pound to Mars. So these costs are substantial, and NASA is always looking for ways to bring that down. And one of the ways to do that is to bring down mass of the spacecraft through creating new and developing new, better materials that are stiffer, stronger, and lighter weight. And doing that, we may be able to improve the performance of the spacecraft and also bring that mass and those launch costs down. So this project went after that by trying to combine 3 different exciting technologies. The first of them is carbon nanotube yarns. So carbon nanotubes have been around for quite some time now and received a lot of research over the past few decades. Their theoretical mechanical properties, stiffness and strength, are off the charts. However, the challenge has always been in scaling them up. So although at the nanoscale they may perform incredibly well, we need meters and meters of material to form a spacecraft, and the properties have never translated up the scales to be realized at those length scales. Manufacturers and researchers have been working really hard on this problem. They've started to create larger structural elements like yarns and CNT sheets, which you can see here are quite large in 2 dimensions, but very thin in the 3rd dimension. And so the challenge is when we have these sort of 1D materials, how do we actually create a 3D structure from them? And the assembly process then dictates the properties. So for example, if we stack those 2D CNT sheets up, even though the sheets are really strong, the weak spot becomes the interface between the sheets, the glue that's kind of holding them together, and that's where material fails. So for example, you may get cracks in between the layers called delamination, and the material fails quite early. So the second piece of this project was to try to assemble these 1D or 2D elements in a different way. And that is 3D weaving. The interesting manufacturing process. I'm sure you're familiar with 2D weaving that's used to make clothing, for example. 3D weaving is a little bit different. We can stack multiple layers of material together, and more importantly, we have this Z yarn that goes in and out of the thickness and really binds that fabric together. So you can see that over here in a metallic woven sample. This yarn that's going up and down really squeezes the material together and will prevent it from opening up and give it much higher strength against this effect of delamination. So Hopkins has a 3D weaving loom, and Tim Weiss's can weave all sorts of different materials, and the goal of this project was really try to do that with CNT yarn and see what we could get. The third piece of this puzzle was topology optimization, which is a computational design method. Topology optimization really combines mathematical optimization and physics-based modeling and tries to arrange material in the most efficient manner. So this is just an example of a beam where we're trying to reduce mass and maximize stiffness, and you can see it creates this sort of organic shape that uses material most efficiently. So we have here a material that serves as our fundamental building block. We're assembling that in a way that takes maximal advantage of the building block's properties. And then, of course, we're using a design tool to guide what we should actually manufacture. So when we received the grant, we got right to work on actually modeling these CNT composites and creating a topology optimization approach to design them. So we can generate those yarn architectures, we can infiltrate them with a composite, we can look at load paths through the material and see how it behaves under load. Our collaborators at NASA who work on CNT materials and have this issue of delamination, really excited by this project. They saw a lot of potential in 3D weaving and were excited by the seed grant, and actually sent us a ½ kilometer of carbon nanotube yarn for us to try to weave. So that was very exciting for us to see NASA buy-in right from the get-go. And Christina got right to work on characterizing that yarn and arrived in January, and she started testing the strength and geometry, make sure it could fit through our reeds and bend to the geometries it needed to. The good news is it passed all those tests, so we had some confidence that it could be 3D woven. The sad news is she was scheduled to start weaving first day of spring break. So that's the kind of student she is. She was super excited about doing research over spring break, which is awesome. But unfortunately, the labs closed, of course, and undergraduates were sent home just a few days before she was about to get started on the actual manufacturing. So that was disappointing. Because of that, we pivoted a bit and went back to computation and looked at another manufacturing process called toe steering. The way to think about this is, yarns and tapes and ribbons can be put down in really straight paths, and that would be simple. But if we have robots that can precisely, then we connect them in curved paths and get different properties. So over here is NASA's Isaac robot, which can do just that. I encourage you to find a YouTube of it. It's a really amazing machine. But we created a design method that then optimizes how these yarns and ribbons and tapes can be aligned, and basically aligning them in the strongest direction or the direction of greatest need for strength, for example. So even using the same material, just putting in a different pattern, we can get really high gains in stiffness and strength. So in terms of sort of steps, really hoping the lab up for her undergrad so Christina can get back in and actually make a sample. NASA is super excited by this, and they've offered not only sent us the yarn, but they've offered to infiltrate the sample with resin and actually perform a structural test on it, as well as now excited about the toe steering results and offered to try to manufacture and test one of those as well. So that's all exciting stuff. We hope the things perform well so that we can increase that collaboration and hopefully lead to new projects. Finally, just some acknowledgments. Thank you very much to Space at Hopkins for the support. It's been great. Thank you to our collaborators, and of course, the project team, and again, the students who really did all the work. If anyone's interested in learning more, just please send me an email. I'll be happy to chat about this project. And thank you very much.
Take it away.
Okay, thank you. So the first question is whether this work, to paraphrase the question, how nature creates high strength materials. Is that an inspiration for the type of work that you are doing?
Great, yeah, thank you for the question. So first off, sorry for those audio glitches. I must have recorded the talk about 10 times and each one had audio glitches, and that probably had the least, so apologies for that. So as far as nature and materials, certainly over evolution, nature has improved the design of structures and materials. And there are some interesting similarities. So some materials, such as nacre, you may have heard of, use soft and tough materials or soft and strong materials together, hard materials together, and stack them in different ways, much like a structural composite could be stacked. So they arrange soft and stiff materials in arrangements that will give you certain properties. We don't do as much of that. We use optimization to guide our design a little bit more. And one of the reasons for that is because nature, in addition to sort of optimizing, for example, strength, they also have to take care of a lot of other functionalities. So for example, in your bone, you may have nutrient transport, you may have repairability, right, self-healing and things like that. So nature often has a lot of other functions in their design. So often it's not a direct one-to-one matching, but certainly in terms of inspiration, nature is a great place to look.
Okay, thank you. Next question. So can you tell us a little bit more about the dimensions of the CNT yarn, and can you elaborate on how you perform the strength and bend testing?
Sure, yeah. So the CNT yarn comes in a lot of different flavors. So part of a NASA program we're involved with is actually focusing on trying to create yarns and create different lengths and different diameters and different strengths of that CNT yarn. The yarns that we have in hand and are working with are about 200 micron in diameter. And again, about half a kilometer in length. One of the sort of unique things for us in dealing with this is we're used to dealing with metallic wires in our 3D weaving loom, which are constant diameter. The CNT yarns have a little bit more variability because certain parts of the yarn, the bundles, you get more bundles in certain parts than others. But that variation wasn't too bad, about 20 or 40 micron variation from that 200 micron diameter. As far as the testing, we did some simple MTS testing, and in terms of bendability, we kind of just ran it through our 3D weaving equipment. So for us, the bendability that's really important is, one, can it get on a spool? So you saw maybe a picture with all those spools on it. Can it be spooled up? And the second one is, can it wrap around the weave? And you saw those wires that kind of bend up and go down. For us, that means it has to do a 400 micron turn relatively tightly, and it was able to do that very easily. We can even tie a knot on itself. It was that flexible.
Okay, thank you. And I think we'll have time for this one last question, which I hope you'll understand what's being asked, because I'm not sure I perfectly do. Can you make the materials multifunctional using your techniques?
Sure, great question. Yeah, so can the material not only be strong, but can it conduct electricity or cool, become a thermal cooling device? Yeah, that's quite a bit of, we do quite a bit of work in our group in that arena, trying to design things to have multiple functions. For example, load bearing, heat exchangers, things that can electrically conduct in a very tailored way and still have some mechanical properties that are needed for the application. So yes, we do quite a bit of that kind of work.
Okay, thank you. That is all the questions we have for now. So with that, I will hand the virtual floor back to Carolina.
Thank you very much, James, and the audience. As a reminder for anyone who is just tuning in, we are using the Q&A feature for questions, and feel free to ask questions during the talk, and Matt will be moderating. So our fourth seed grant speaker today will be Dr. Rachel Austin. Dr. Austin is an associate research scientist in the Department of Physics and Astronomy in the Krieger School of Arts and Sciences. She's also an associate astronomer at the Space Telescope Science Institute, where she serves as deputy mission head for the Hubble Space Telescope. She is a multi-wavelength astronomer who specializes in the dynamics of cool stellar outer atmospheres.
Okay, thank you very much for being here and listening to the results of these seed grant proposals. I'm here to talk about a project that I've been working on with Professor Colin Norman in the Physics and Astronomy Department and Dr. Angelos Forlidis at the JHU Applied Physics Lab, where we're looking at new tools to study transient stellar mass loss. The motivation for this project stems from the recognition that we now know of thousands of planets outside of our solar system. And in order to have a more comprehensive understanding of the types of environments those planets experience, we really need to understand the impact that the host star can have on those environments. And this was recognized in this 2015 NASA astrobiology strategy study that asked the question, what properties of the host star are conducive to or prevent the formation of a habitable world? So that's a recognition that stars impact their environment. So for stars like our sun that are about the temperature, mass, radius of our sun and cooler, we know that those stars have magnetic fields that thread the surfaces of the stars and lead to magnetic activity, in particular to flares, coronal mass ejections, and energetic particles. The image on the left is a combination of several different images of the sun. The innermost one that's red looks at the atmosphere of the sun that's near the surface. This is the so-called coronal material. And the two green images show solar emission that is further removed from the sun. And in particular, you can see in the bottom right that there is a large shape of material that's leaving the solar environment. This is a coronal mass ejection that happens with flares. These cause space weather in our solar system. Around more extreme stars, these coronal mass ejections and energetic particles can result in a threat to the potential habitability of planets. Now, we can see stellar flares on other stars easily. And we know that the most active stars can have flares that are 10,000 times more energetic than the largest solar flares. But we don't have a ready method to detect and diagnose coronal mass ejections in stars. And this problem is compounded by the fact that when we observe the sun, as in this sequence of images on the left, we can actually see the surface. We can study it. We can study the sun by looking at it as a function of space, as a function of time, and as a function of wavelength. When we look at stars, even though we can make incredibly detailed artist conceptions of these stars and what their stellar flares and coronal mass ejections might look like, we cannot actually resolve them in space. So we can only use those other two parameters, breaking up the light of the star in wavelength and studying how that changes with time. So we have to use tools that enable us to tease out these inferences from those two methods. And so so all of that is just a way of saying we can't necessarily take all of the observational tools that have been developed for the sun and apply them to other stars. So we have to come up with new tools. So this tool that we are investigating is something called absorption dimming. This was suggested in a paper several years ago by a solar physicist that if the specific region on the surface of the star that's producing the flaring is covered by another material that's being expelled, as in this sketch here on the left, then that could cause a temporary increase in the amount of absorption that you're seeing from the integrated system. So even though we can't spatially resolve the surface of the star, we can use the time-dependent changes in properties to infer that this situation might be happening. This image on the right is a result from a previous study that interpreted this time-dependent change in the amount of the absorbing column density during a large flare as evidence for a coronal mass ejection. And we want to test the utility of this tool for detecting and characterizing stellar coronal mass ejections. We're using as an initial sample the largest stellar X-ray flares that have yet been seen with the Swift Space Telescope. We're currently in the data analysis and interpretation phase for the most promising candidate right now. So things got a little bit delayed with coronavirus, but we're hoping that we'll have some preliminary results in the next few weeks, sufficient to motivate a larger study of this entire data set, and then an expansion to other X-ray samples of stars.
All right, thank you. Go ahead with the Q&A.
Okay. Pardon me one moment. So the first question is whether there are other sources of dimming or brightening that you would potentially have to extract as contaminants from your observations that might mimic the sort of signal that you are looking for.
Hi, can you hear me? Just want to do sound check. Okay, great. Before I answer the question, I just wanted to take a few seconds to thank the Space at Hopkins folks for providing some of the seed funding for this project. The undergraduate who worked on this project is in attendance, so I'm going to give a shout out to Sam Salander, who did an absolutely fantastic job, particularly under the circumstances, but even without the circumstances, he was great. So the question was whether there are other things that might, other physical processes that might produce this signature instead of the coronal mass ejection interpretation. And so the stars that we're studying are far enough away from us that we do have to take into account the fact that there's an interstellar medium. There's material between us and the star. So that's an additional layer of absorption, but that is not going to change with time. And that's the key parameter here, that we're looking for a significant change in the amount of dimming with time, which really is hard to explain with any other hypothesis because it requires a very large amount of mass to be moving across the line of sight and produce this dimming. And we can actually take our some simple models for what is happening closer to the surface of the star, and then connect that with our inference of what we think might be happening under this CME hypothesis, and see if those make sense, and then we can connect that back with what we understand about the sun, and see if that also makes sense from a physics perspective.
Okay, thank you. And another question: can you say a bit more about the specific types of stars that you're studying to try to develop this new tool, and what sort of the extension of the concept of habitability and systems like that might look like?
Yeah. So in order to study this process, we have to look at the most extreme types of stars in terms of their magnetic activity. And so we're looking at generally very young stars, red dwarf stars, which are known to produce frequent and extreme levels of magnetic activity, and some stars that have a companion. So there is overlap between those stars and the kinds of stars that we're finding planets around. We're finding planets around young stars and M dwarfs. It's sort of an interesting region of parameter space to study this phenomenon at the extreme, and then understand the physical processes that are producing it, so that we can then look at, I mentioned that there's a gap of about 4 orders of magnitude between what we see on the sun and what we can see in these extreme stars, and we want to have an understanding that the physics scales, right? And so if it does, if we can show that that's the case, then you can look at more moderately active or even inactive active stars as planet hosts and have a better understanding of what might be happening with them.
Okay, thank you. That is all the questions that we have for now. So with that, I will pass the virtual stage back to Carolina.
Thank you, Rachel. So our 5th and final seed grant speaker today is Professor Trina Schroer. Professor Schroer is a professor in the Department of Biology in the Krieger School of Arts and Sciences. Her research addresses questions of cell motility. which is movement within the cell and movement of the cell as a whole. She is an expert in microtubule-based motor proteins and their roles in cellular architecture.
Hi, I'm Trina Schroer, and today what I want to do is tell you about some work we're doing to try to develop genetic screening methods for determining whether or not potential astronauts might be predisposed to cataract formation, which is a common and well-known thing that happens to astronauts on long-term space missions. So I'd like to start by just briefly reminding you about how the eye lens works. Basically, light comes in from the front of the eye and is projected through the lens to the back of the eye. And to do this, the lens requires a perfect optical fiber that's perfectly transparent and maintained through the life of the organism. And that's these cells here in the center of the lens. And so these make just this perfect fiber. And the way these cells are organized into this structure and maintained is that they make very, very tight contacts with cells in this epithelial layer. And these cells have a structure called a cilium, which is important for patterning. So defects of this lens structure or the interaction of these two cell types can lead to defects in clarity. My lab studies basically very, very important housekeeping protein that's critical for cell function called dynactin. And that's this colorful thing here. And what dynactin does is it's an accessory factor for an essential motor, dynein, that's responsible for moving a vast array of material around inside the cell. So here are zoomed in views of this P25 protein here. That's the topic of the talk. It's shown here in turquoise, here in blue. And in fact, we're going to focus in even deeper and just talk about the function of just this one tiny little portion of this P25 protein. So loss of just this little piece of P25 in this whole dynactin molecule makes animals unable to survive and can cause lens clarity defects. So we stumbled upon this because someone in another lab identified in a mutant screen a mutant protein, a mutant form of our protein P25. Animals that are homozygous for this mutant gene don't survive, but luckily for us, heterozygotes are viable and fertile, so we could study them further. Now, fortuitously, this truncated form in this mutant is essentially identical to a naturally occurring splice form that's expressed in lens. And so this has some interesting implications for how this protein contributes to lens function under normal circumstances. What we found in heterozygotes is that they do have some interesting lens defects, and then I'll show you that work first, and then also I'll very briefly show you that at the cellular level we've shown two specific defects that may be responsible for these lens clarity defects. So what we did was you take the lenses from these animals and you can just look at them in the microscope. And the first thing we did was just see how well they transmit light. And so to do that, you put the lens on an electron microscopy grid. The lens is a lens, and so it magnifies. And so you see here a perfect magnification of the hexagonal pattern of the grid. But when we took lenses from these mutant animals, what we found was that they showed very obvious defects in this pattern. So that suggests that the cells in those fiber cells are not perfectly patterned, or they're not perfectly structured, or something else. We also observed defects, an interesting defect, which was when we put these lenses at slightly reduced temperature, the central core of the lens, which is supposed to remain clear, precipitated. And this is intriguing because this phenomenon, which is known as cold cataract formation, is typically associated with cataract formation later in life in other animal models. So we then moved out of the mouse into cultured cells where we have more ready access to genetic manipulations, and looked at the behavior of some proteins that are important for cell organization. One of these is actin, labeled with a fluorescent probe. This is what the actin in normal cells looks like. You can see ruffling at the edges and these fibers. And that's in dramatic contrast to what cells that have been depleted of P25 by RNA interference show very, very different behavior. When we add normal P25 back, we're able to restore this cortical behavior. The cells are much less ruffly. But when we added back the truncated form in place of the wild-type normal form, we again see defects in the cell cortex behavior. So that clearly shows that just the loss of that little tiny tail on the P25 protein is responsible for this defect. So for the SEED grant project, basically what we did was extend these findings and look at a couple of other even further truncated forms that are present. Isoform 2 is another splice form that creates a very, very small form of P25. It has unknown function. And then we also explored the behavior of a naturally occurring human variant form that's present in about 1% of the population. And so basically, these two forms that we looked at with seed funding would encode a protein that was a very, very small piece of P25, just probably the first 2 or 3 wraps here. So much, much smaller than the entire P25 protein. So what we wanted to do with seed funding was to characterize these proteins. Are they made? Can they assemble into dynactin? And then if they do, which we didn't know if they would, do they give similar defects to dynactin? And the bottom line is that these proteins are so small that they do not seem to yield stable protein. And because there's no stable protein, then we don't actually see, these cells have exactly the same defects as cells that lack P25 altogether. So just briefly, let me walk you through this. We were not able to measure the really, really small truncated forms of P25 because we didn't have appropriate probes. So we looked at this by proxy. P25 normally is bound to this protein here, P27. The two come and go together. If you deplete one, the other one leaves. If you replace one, the other one comes back. And so that's shown here. And with the form that just lacks this short structure here, the truncated form, NEMO, we do get protein back, and this does rescue P27 protein. But as you can see, neither of the other truncated forms yield any P27 protein. So that suggests that there's no protein made. Just to find out whether or not these are actually, even making RNA, we went ahead and looked at that. And yes, in fact, all of these forms make RNA. So they're making the transcript, they're just not yielding stable protein. So basically what we have is a situation where these completely lack P25. We looked at cilium formation, which is one of our in vivo readouts, and we found that these truncated forms behaved, basically exactly like cells completely lacking P25. So it's as if these are not made at all. So that's about it. Basically then, the take-home message here is that we do have a screening tool for looking at predisposition to cataract and potentially other degenerative phenomena. And this has some interesting impacts for future sort of selection of crews for space missions, et cetera. I just want to acknowledge Ting-Yu Ye, who did all the work here, some undergrads who assisted with the project, collaborators here and at Delaware, and of course, thanks to Space at Hopkins for the seed funding. Thank you very much.
All right, thank you. You can go ahead with the Q&A.
Okay, thank you. So the first question is a clarification, if you're able to share your screen, Professor Shore. On the last slide that you showed before the acknowledgements, on the left side, can you go back and tell, for those of us without strong biology backgrounds, what type of measurements are being made in the figure that's a gray rectangle with the black lines for the different proteins? You're muted at the moment. We can't hear you yet.
There we go. How's that? All right, let me pull up my PowerPoint. So you said the last slide with the gray. Right. The gray lines, yeah, the data slide.
Exactly.
The deadly data slide. Okay, share my screen. Here we go. So what these are, each of these bands, so what we do in this experiment is to electrophorese, so subject proteins to an electric field, and then they migrate according to size. And so these bands are basically an indication that there was a protein in that sample. And then the way that we selectively visualize just that specific protein is we use an antibody, which we've been hearing a lot about antibodies lately. And so antibodies to different proteins will then, if they yield a black band, then that means that there is the protein there. And if it's blank, then that means the protein is not there. So I'm just going to hide this floating meaning control. So what I didn't explain as clearly as I might have was that I tried to make the point that these two proteins always come and go together. And so here's a case where they're both present, and then here's a case where we've depleted one of them and then they both go away. And so you're just looking for this pattern of is there one band? And if so, then that tells us that this protein down here that would be in this part of the sample is not present. Did that help?
Yes, indeed. That definitely makes the figure more comprehensible.
Yeah,
Thank you.
It's very technical, and it's so there, I have to make some assumptions, but I try to do my best there.
Thank you. Well, now I hope this doesn't throw you too far off balance, but the next question is going in completely other direction. So as I understand it, the relevance of this research to the human spaceflight is that cataracts are a problem that astronauts may experience on spaceflight. And so you're looking at the possibility of something that could predispose them to be either more vulnerable or more resistant to forming cataracts.
Yes. Oh, sorry. Go on.
Well, thank you for confirming that understanding is correct. So the bigger question is, eye problems are something that we hear about astronauts having. Joint problems, we heard an earlier talk about the type of fatigue that can develop and the damage that can develop. Do we need to be thinking in terms of there being many, many different individual systems of the body which would have to be screened against and selected for in order to create an astronaut or to find an astronaut who would be well suited for a long duration spaceflight? Or what can you tell us about that?
Well, I think that, the genomic era of biomedicine places us in a position where, very, very specific risk factors can be identified and screened for. And just to sort of out of the realm of space, but into the realm of COVID, right? Scientists are looking for, what type of gene, backgrounds might make a person more susceptible to get severe disease. And that really is where medicine is going. And my first slide, am I still sharing? Because I've completely lost all my controls here.
Yes, I can see it.
You can see this, and I can make this go away. So basically, I always wondered in Star Trek what they were doing with those tricorders. But what they're doing is they're reading the chip in your neck that has your complete genome information on it, so that they can tell you exactly how to precisely prescribe the right kind of medicine that will help you with your genetic background. And this is where medicine is going. And I think that in terms of astronaut health, one of the human research roadmap that NASA is developing is trying to identify radiation protective pharmaceutical interventions. And my feeling is that we need to also start by understanding the body itself and sort of thinking intelligently about what sort of risk factors might be an issue, right? So maybe, osteoarthritis is not good, terrestrially, but in a person who's going to be out in space, maybe, there might be something about that could actually be in favor of health. And so I think that the short answer to your question is yes, there are definitely lots of systems. And NASA is trying to, bring together scientists who will really be able to provide a comprehensive picture of, what is it that's involved in cardiac degeneration? Why is it that this subset of astronauts had an effect and others didn't? And so I do think that this is moving in the direction of pre-selection. But taking people who just have family histories that sort of lack degenerative disease would suggest that those people are probably going to be less susceptible to degenerative effects of radiation. So just as an example.
Okay, thank you very much for that answer. I think that is the last question that we have for the moment. So with that, I will hand it back to Carolina.
Thank you, Trina.
Thank you.
Thank you once again to all of the seed grant speakers, and congratulations on your very successful seed grant projects. We encourage anyone with further questions to reach out directly to the seed grant speakers in the days ahead, and reach out to me if you're curious about seed grant research or awards. I'd like to now introduce our final speaker, Baltimore native and Johns Hopkins University alumnus, astronaut Reid Wiseman. It's my great honor to be able to introduce his talk today. Although he could not be here live today due to his busy astronaut activities, astronaut Wiseman has generously provided us with a special talk that I am absolutely thrilled to share. Astronaut Wiseman grew up just around the corner. He attended high school in Symposium, and he earned his Master of Science degree here at Johns Hopkins in 2006, under the Whiting School of Engineering program in Systems Engineering. His impressive career and contributions to space research are an excellent example of what a Johns Hopkins alumnus can achieve. Astronaut Wiseman was the flight engineer aboard the International Space Station for Expedition 41, a 165-day mission lasting from May through November of 2014. He also commanded the NEMO mission at the Aquarius Reef Habitat off Isla Mirada, Florida. Aboard the ISS, astronaut Wiseman and his crew completed 2 spacewalks and over 300 hours of scientific experiments, spanning research areas including human physiology, medicine, physical science, earth science, and astrophysics. In July of 2014, astronaut Wiseman and his crew set a record aboard the ISS of 82 hours of research in a single week. So without further delay, I will let astronaut Wiseman take the virtual stage.
Hey, good afternoon, everybody at SpaceSat Hopkins. Wish I could be there in person. If everything's going as planned right now, I'm on a NASA aircraft coming back from Kazakhstan, and I just went over there to pick up our most recently returning astronaut, Chris Cassidy, Naval Academy graduate from just up the road. I want to start by thanking Dr. Bennett for putting this on and getting everyone together, uniting researchers and students in the common pursuit of civilian space research. From my perspective as an astronaut, I really can't think of any greater cause right now than that. What I'd like to do over the next 20 minutes is just talk through a little bit of civilian space research from my perspective, from a NASA astronaut perspective who was on the space station for about 6 months and came back. I thought the best way to do that is, first, I'd like to take you through launch, get you up to the space station, and then let you feel kind of what it feels like to live and work up there on this magnificent national laboratory that we have in low Earth orbit. Then I'd like to show a couple of pictures from space, and then go through where I think we are today in civilian space, and then where I think we're going in the very near future. So with that, I think we should climb aboard a Russian Soyuz rocket and leave the planet. My launch was in the middle of the night, early in the morning. You walk out, you get all dressed out in your spacesuit, say goodbye to your kids and your family, and then you're heading out to the rocket. For me, saying goodbye to the kids, that's the hardest thing possible to do, but you know that rocket is waiting for you and you want to get out there and get onto space. So I'm out there with the Russian cosmonaut Maxim Sarayev, German astronaut Alexander Gerst, and myself. We get out to the rocket, and that thing is alive. It's completely fueled. It's covered in ice. There's steam coming off of it. It's creaking, and it's pretty scary, but it's pretty inspiring at the same time. So you ride the elevator up, you climb into the capsule, and then you wait about 2 hours before it's time for liftoff so they can clear out the pad. When it's finally time to go, those engines light, and you know you're going somewhere and you're going fast. So it's a 9-minute ride, about 9 minutes to get from Earth into low Earth orbit. And in that 9 minutes, you go from 0 miles an hour to 17,000 miles an hour. So it is an incredible, awesome experience. There's a lot of G-forces on the body, and the ride is really it's actually pretty wonderful. So nine and a half minutes, and you're going 17,000 miles an hour. And it's the Russian Soyuz rocket is a 3-stage rocket, and those engines are burning the whole way up. When you finally make it into space, this was my first flight, and those engines cut off, and you're floating in low Earth orbit, there is this total sense of euphoria that comes across the entire crew. I don't care how many times you've flown in space, you just feel amazing when those engines cut off and you're safely up there. we had a little camera in the cabin with us, and I just remember reaching up and pulling that thing off the wall, and this is my first view out the window. It is incredible. No camera does that view of our home planet justice. It just takes your breath away. So now that we're up on the space station and we've docked, it's time to come inside this national laboratory. It's an enormous facility, and let's just take a little day in the life of an astronaut cruising around the space station. So first few days that you get up there, you feel pretty sick. At least I did. And then after about day 4, I started to really feel fantastic. At first, you're clumsy. You may have a headache. You may feel like you're nauseous. Your body really doesn't want to go to the bathroom, really doesn't want to eat. But after a few days, you really settle into a nice rhythm. And then I was up there for 165 days, almost 6 months, going 17,500 miles an hour, cruising around the Earth once every 90 minutes, 16 sunrises, 16 sunsets every day. But life inside this machine is pretty easy. It's pretty fantastic. You're floating around. You got window views of the Earth, which I'll touch on in a minute. But our primary goal up there, our objective for being on this national laboratory is the science. And there at Johns Hopkins, at the Applied Physics Lab and the Whiting School of Engineering, I know you all are pretty deeply rooted in the science that we're doing up on the space station. When I was up there in 2014, the big research that I was working on was bone loss, epigenetics, sensory adaptation. What is radiation doing? Your bones change, your blood volume changes, your heart changes, your eyes change. Everything inside the human body changes. So every single week I was taking blood, taking tests, doing ultrasounds, looking at the retina, looking at the backs of my eyes, seeing what really is going on with the human body over a long duration spaceflight. We're just scratching the surface of what is going on with the human while we're up there. At the same time, we got to do maintenance to keep the space station running, so a lot of our day is doing either preventative or maintenance when something breaks. We got to take care of our body, so running on the treadmill, lifting weights, those are huge parts of our day. And then, of course, we get about 10 hours off a day, and they usually try to give us Saturdays and Sundays, light days at least. And those, for me personally, were some of my favorite moments. That's when you get to float over to the window, maybe have a nice, relaxing Sunday breakfast, go down to the cupola, which is a great viewpoint on the Earth, and we get to look outside at our home planet. Every single time I look down at Earth, I wanted to reach immediately for a camera. There is nothing more beautiful than looking down at our home planet. It doesn't matter if you're looking down on a hurricane, if you're looking down at Aurora, if you're looking down at your hometown of Baltimore, Maryland. This is a great shot of Maryland during the day. You can see the Chesapeake Bay. You can see how green and lush Maryland looks from space and then out to the Atlantic Ocean. But what really blew me away was looking down at Maryland at night. I could look at this picture for the next half hour easily. You can see the I-95 corridor basically running from New York through Baltimore, down through DC, and then on to the south. And remember, this picture is just looking through one window. So looking out the back of the space station right here, you can follow 95 all the way down to Miami. You can kind of make out the Chesapeake Bay, you can see Philadelphia, and if you look off in the distance towards the horizon, you can even see parts of the Great Lakes up into Canada. You can see Boston off on the coast. You can see Detroit. You can even peek and make out Chicago way over on the side. Just looking down at that and knowing how many millions of people are down there, it really blows your mind. So for me, my reflecting moments were always trying to get to a window and looking back at our home planet. At the end of that 6 months, it's time to come home. The ride home it's a lot of fire, it's a lot of G-forces, but it's actually relatively short. And you don't have a lot of say in the ride down. You hit the brakes, start decelerating towards the atmosphere, and as soon as you start hitting the atmosphere and the friction builds up, the G-forces build up, the capsule gets very hot. These views out the window are basically fire and plasma going by my head about a foot or two away. It's pretty wild. And then at the end of all that, parachute comes out and you float down to the steppes of Kazakhstan, and rescue forces are right there, ready to pull you out of the capsule. We waited maybe 30 seconds after we landed, and the rescue forces were already on us in the desert. And then it's time to come home and get reunited with your family. So that's 6 months in space in a nutshell, and I hope that just leaves you with a tiny perspective of when you're sitting on the ground in a lab looking at what we're doing in space, maybe just a little shot of that will be in your brain from now on, and you can think about what it would feel like to be up there floating with us. I hope soon every human being gets a chance to do that because it's completely worth it. The space station really has become this true laboratory where we are conducting government research and civilian research side by side every day. We have completely private companies like Cataract. When I was up there, I did an experiment with Procter & Gamble. 5% of our time is dedicated completely to private industry and carrying out their research, and they can use that time for whatever they want to. We also have what we call KSAS, the Center for Advancement of Science and Space, and that is really streamlining the way civilian companies and personal researchers can get their payloads up on the space station. In the time I was up there, our crew did over 300 experiments, and I would say probably half of those were not government-based experiments. That was running the gamut of anybody that wants their research done. The door is open. Business is open on the International Space Station to get that research done. So now that we're back from space, I'd like to take a step back in my own life, in my personal journey of how I got there, and just touch on a few key points. I went to college up in New York. I grew up in Maryland, just up the street from everybody at Johns Hopkins, just north of Baltimore in a little town called Cockeysville. Went to college up at Rensselaer Polytechnic Institute in New York. I studied computer engineering. And after I finished college, I decided I really wanted to fly. Like, that was what was in my blood. So I joined the Navy out of college, and I was very fortunate, got to fly F-14s and F-18s off of aircraft carriers. That was a magnificent job. As part of that, I did a tour at Patuxent River, Maryland, which is just down the street from y'all at Hopkins. And I took about 3 years. I became a test pilot with the Navy. And as part of that, I found this little program through the School of Engineering there at Johns Hopkins to study systems engineering for a master's degree. And that was how I got connected to the great things that you're doing at Hopkins with the Applied Physics Lab and the School of Engineering there. So for 2 years, I attended night school, and I went through the systems engineering degree. Essentially, I learned everything I needed to be an effective astronaut at that school from an academic sense. I learned everything about schedules, how to put together a program, how to work with a program manager, and that small education, that 2 years, has proved to be some of the most useful education that I have had in my entire life. After I was a test pilot in the Navy there at Patuxent River, I moved back to Virginia Beach, and in 2009, I was picked up by NASA to become an astronaut. So I wanted to piece in that little Johns Hopkins part just so you see, like, that little tiny decision for me when I was at Patuxent River to take that little extra time and get a master's degree through Hopkins wasn't necessarily easy. It wasn't a little bit of time, but it turned out to be fortuitous. It was really one of the best decisions that I made. So now let's just take a quick glimpse at where civilian space is now. Probably most of this is review for you if you're in a space conference, but I just returned recently from Cape Canaveral, Florida. I was down there for a quick training event. And in my time just walking around Cape Canaveral, I saw 3 rockets on the pad, a Delta Heavy and 2 rockets from SpaceX. Right now, SpaceX is launching cargo to the International Space Station. It's launching crew to the International Space Station. It's launching the Starlink satellite network. It's launching GPS satellites. And of course, in South Texas, they're working on the Starship, which will eventually go to the moon, Mars, and maybe beyond. That's all SpaceX. We also have Northrop Grumman. They are launching the Antares spacecraft just down the road from you all at the Wallops Island facility off the coast of Maryland and Virginia there on the eastern shore. So they're launching a cargo ship up to the space station right now, Northrop Grumman 14, a Cygnus spacecraft. We also have Boeing working on crew capsules. Boeing is working on satellites, Defense Department research. We have Blue Origin, which is doing suborbital spacecraft, soon to have humans on board. Virgin Galactic has already flown humans into suborbital space. We have loads of balloon research. Another company coming online called Axiom is selling tickets to the International Space Station. So maybe some people have heard about a pretty famous actor that's supposed to fly soon. I don't know if it's true or not, but that'll be amazing. That is opening up the chances for everyone on Earth to get off our planet. And then we also have companies like Rocket Labs and Relativity Space. And these are the smaller startups, but they're doing amazing things. The reason I wanted to bring them up is we have an entire commercial industry that is on the brink of just taking over at least low Earth orbit, suborbital and low Earth orbit. We are at a spot that we have never been in our lives before. And certainly, if you're researchers trying to get your payloads up suborbital or orbital flight, it used to be one game in town, and now you've got 20. And that is today. All of that I just talked about is flying today or in 2021. That is amazing. And then from the NASA perspective, if we want to look just a tiny bit into the future, towards the end of 2021, we're going to be launching our first Artemis mission, Artemis 1. It's going to be an uncrewed test of the Orion spacecraft and the Space Launch System. It'll spend about 44 days out in lunar orbit and then come back to test out all those systems that we need to make sure work before we put crew on Artemis 2 in 2023. When I was at Cape Canaveral, I was looking right at the Artemis 2 spacecraft. It's about halfway outfitted, and the thoughts were just going through my mind of, man, this capsule right here is taking us out to the moon, taking humans back to the moon, and then on Artemis 3, we hope to land on the moon in 2024. We are not going to land on the moon without civilian space. So right now we have contracts out with Dynetics, Blue Origin, and SpaceX. All three are simultaneously working on building lunar landers that will take a crew of 2 to the lunar surface in 2024. And that's just the tip of the iceberg. That's a test flight to try out these systems. And then from there, we're going to be taking 4 astronauts to the surface. We're going to be building habitats. We're going to learn how to use the resources on the moon to fuel up rockets to get us home and to get us onto Mars. That's really the goal for NASA right now, is to get to Mars. But looking beyond human spaceflight, Venus, amazing discoveries. Is there life on Venus? We've got to find out. I know you all know that we're sending Dragonfly out to Titan. We've got the Europa Clipper to go try to look at what is going on these moons in Jupiter and Saturn. And then the end of human spaceflight is not our solar system. How do we get outside of our solar system with crew on board? And there's companies like Limitless Space, a nonprofit down in Houston, looking at warp drives and how do we do solar sails and just anything you can possibly imagine to push those limits, to make us limitless and send humans outside the solar system. So looking back on everything that's going on right now, it's amazing. You can't look anywhere and not see civilian space absolutely taking over. And the world is our opportunity right now. There's never been a better time. It's the golden age of civilian space. I think everybody watching today has a chance to leave our planet on a spacecraft and go check out what does our Earth look like from space, and I highly recommend it. So in closing, thank you so much for giving me the opportunity. Again, I wish I was there in person, and hope to meet all of you someday soon.
And as we leave the moon, we leave as we came, and God willing, as we shall return.
You probably dreamed of this.
Liftoff on Apollo 11.
You probably replayed this moment 1,000 times.
4 wheels off the ground air.
You probably imagined the awe-inspiring, indescribable rush of the ride. You probably celebrated the miraculous, cheered the momentous, and mourned the shocking. You probably still dream of what could be. We did. We do. And while many thought we were through, no. We've been learning, we've been building, we've been training. And now, it's time. Because the next frontier is not just for the next generation. It's for this generation. It's for me, and it's for you. For everything we still dream about. We go as Artemis.
This concludes our event today. I would like to thank all of our panelists for the exciting and inspiring glimpse they have provided into space research at Johns Hopkins. And thank you to all of our attendees for calling in and participating with us here today. Once again, we encourage you to be in touch with each other. If you have any lingering questions, found the research to be exciting, enjoyed a presentation, please don't hesitate to reach out. Information about Space at Hopkins, Seed Grant awards and recipients, and past events can be found on our website, spacestudies.jhu.edu. I am the primary contact for Space at Hopkins, and you can reach me at spacestudies@jhu.edu if you have any questions. If you have not yet done so, please join Space at Hopkins at our website to get on our email list for notifications of future events. I'd like to thank the president of the university, Ron Daniels, and the provost, Sunil Kumar, for their continued support of the Space at Hopkins program. Lastly, I'd like to personally thank Professor Bennett, Matt Collins, and John Schroeder for your tremendous support, assistance, and vision in bringing this event to fruition. Thank you, and I look forward to seeing you all, hopefully in person, at the next Space at Hopkins event. Thank you.